Photosynthesis is the process by which green plants, algae, and some bacteria convert light energy into chemical energy stored in glucose molecules, while also producing oxygen as a byproduct. This vital process occurs in specialized organelles called chloroplasts, primarily in the leaves of plants. The overall equation for photosynthesis can be represented as 6CO2 plus 6H2O plus light energy, C6H12O6 plus 6O2. This equation illustrates the conversion of carbon dioxide CO2 and water H2O into glucose C6H12O6 and oxygen O2, powered by light energy. Photosynthesis consists of two main stages: the light-dependent reactions and the light-independent reactions Calvin cycle. 1. Light-dependent reactions. These reactions occur in the thylakoid membranes of chloroplasts and require light energy to take place. Here's how it works. Light energy is absorbed by chlorophyll and other pigments in the thylakoid membranes. This absorbed energy excites electrons within chlorophyll molecules, causing them to enter a higher energy state and eventually be transferred to electron carriers. As electrons move through the electron transport chain etc., energy is released and used to pump protons H+ from the stroma into the thylakoid space, creating a proton gradient. The flow of protons back into the stroma through ATP synthase drives the synthesis of ATP, a process known as chemiosmosis. Meanwhile, the energized electrons from photosystem II are transferred to photosystem I via the ETC. In photosystem I, another photon of light energy is absorbed, exciting electrons once again. These high-energy electrons are then transferred to another electron carrier, and ultimately used to reduce an ADP plus to an ADPH, which will be utilized in the Calvin cycle. 2. Calvin cycle light-independent reactions. The Calvin cycle takes place in the stroma of chloroplasts and does not directly require light. It involves several steps to convert carbon dioxide into glucose. Carbon fixation. Carbon dioxide from the atmosphere is combined with a 5-carbon compound, ribulose bisphosphate RUB, with the help of the enzyme Rubisco. This results in the formation of a highly unstable 6-carbon compound, which immediately breaks down into 2 molecules of 3-phosphoglycerate, 3-PGA. Reduction phase. ATP and an ADPH generated during the light-dependent reactions are used to convert 3-PGA into glyceraldehyde 3-phosphate G3P, a 3-carbon sugar. Regeneration of RUB. Some of the G3P molecules produced are used to regenerate RUB through a series of reactions, ensuring the continuation of the cycle. Exit of glucose. While most G3P molecules are recycled to regenerate RUB, some are used to synthesize glucose and other organic compounds, which can be stored for later use or used as building blocks for cellular structures.